Now we come to the experimental part of the geology practical curriculum. The first experiment that we have decided to undertake is the preparation of and observation of a temporary stained blood smear of human. This is the first experiment, and there will be demonstration. Uh, let's have an idea on its principle, how we undertake the experiment. Human blood is a fluid connective tissue. Everybody knows about it. It contains a fluid matrix, the plasma, in which are suspended the three types of cells or corpuscles, namely red blood corpuscle or erythrocytes, white blood corpuscles or leukocytes, and blood platelets or thrombocytes. These are the three corpuscles or cells which are present suspended in the plasma of the blood. A thin smear, how do we observe it? We first of all prepare a thin film on a microslide, then we stain it with an appropriate stain to develop a contrast, and then we observe it under a microscope, a compound microscope. Now, then what are the materials required for this experiment? Requirements: one, a few microslides; two, a disposable hypodermic needle; three, sterilized cotton; four, rectified spirit; five, ethanol; six, Leishman's stain; seven, glycerol; eight, cover glass; and nine, a compound microscope with a mechanical stage fitting. A microscope with a mechanical stage fitting, I'll be explaining when I'll be discussing about its structure. What is seen actually on this slide. Then procedure. Procedure refers to what work we undertake in a sequence, one after another. First is the ring fingertip of the left hand. Look at this. The ring fingertip of the left hand. This is the ring fingertip of the left hand. Is rubbed with an alcohol or spirit soaked cotton. To make it sterile or to make it germ free, you rub it thoroughly with spirit soaked cotton for just a few seconds. Then let it dry. You dry it up. The tip is pricked with a disposable hypodermic needle and then squeezed. Okay. So first step is sterilize the fingertip, ring fingertip of the left hand with rectified spirit. Then dry it up. Then take a disposable hypodermic needle, prick it gently. Then squeeze it. A small drop will ooze out. Then hold the two slides. Not of the slide, please. See, these are the two. Hold two slides like this. You have squeezed your fingertip. Little amount of blood is taken here, like this. Little amount of blood is taken at the edge of one of the two slides. Then this slide, where the blood is touched to another slide at an angle of 45 degrees. So make an angle of 45. Approximately, it's not necessary that you measure the angle, but approximately make it 45 degrees. And see that the blood attached to the slide edge here spreads homogeneously, uniformly on the second slide. Then you draw the first slide on the second slide like this, from here to the end, in one direction only, in a uniform rapid motion. So in doing so, what happens? The blood that is attached here is adhered onto the surface of the second slide as a film. So here you will have this film. This is the first slide, this is the second slide, and the second slide will have the blood film. The movement of the first slide on the second slide is most important. How you move the slide. It must be uniform and very quick or rapid. In doing so, a uniform or homogeneous blood film is drawn on this slide. Okay? Once the film is drawn, you dry it up. You dry it up. After drying, you add only a few drops of absolute alcohol. You add a few drops of absolute alcohol here on the blood film. Wait for a minute or two. The absolute alcohol will dry up automatically. Then the slide is ready for staining. You stain it. There are two methods of staining. One is we may use the conventional method of aqueous hematoxylin and alcoholic eosin. This is one method, the conventional method. And the most convenient method is Leishman's staining. One is conventional staining, the old method, and the other is convenient staining, the new method, the better method. The better method is Leishman's staining method. Why? In the conventional method, that is hematoxylin and eosin, we have to use two stains in succession, one after another. First of all, we stain the slide with hematoxylin, aqueous hematoxylin, remember? Then we stain it with alcoholic eosin. Then we prepare a mount. Then we observe under a microscope. However, in Leishman staining, the single stain will be sufficient for both the stains. You need not have to use two stains. The single stain, Leishman stain, will have the property of both hematoxylin and eosin. We are using a single stain. Add a few drops of Leishman stain. Wait for 5 minutes. After adding Leishman stain, wait for 5 minutes. After 5 minutes, you wash the slide of any excess of stain. Then you put a drop of glycerol at the center of the slide, and next you put a very thin cover glass on it. See that no bubble enters into the mount slide. No air bubble enters into the slide. It's a very easy process. It's not as difficult as many experiments are. It's a very simple experiment. But the difficulty lies in the making of the smear. That is why it is considered sometimes as difficult. Many students are paid to make a smear. A homogeneous smear can only be made when you draw the smear in a uniform rapid motion, or else there will be a break at points and the smear will be heterogeneous, or else it's very simple. So I repeat it once again. Look at this. These are the two slides. This is the position. This slide is with the blood, and this is touched to the second slide. There is homogeneous spreading of the blood on the second slide. Then the first slide is moved forward rapidly and uniformly. So they are very homogeneous, thin and homogeneous granules form. It's very simple. Anyway, so what are the observations? I have described all these states here in the writer. Then observation, what you see actually. Prior to that, let me tell you about a mechanical stage fitting of a microscope. Hopefully, you must have seen a compound microscope, all of you. It's not a very new thing. It has become a very common thing. You have a stage in a compound microscope on which you place this slide. When you view something on the slide, you have to move the slide with your hand. You cannot precisely move the slide. So, suppose you are intending to see something in a focus, you move the slide. So, when you intend to see a specific place on the slide, a specific structure on the slide, you have to move with your hand. It may not be precise. So, to make it more precise, some microscopes have mechanical stage fitting. Mechanical stage fitting enables someone to move the slide in a precise manner, top to bottom, right to left, as he or she wants. This is called a mechanical stage fitting. Anyway, many microscopes do not possess mechanical stage fitting. A simple microscope or a stage without a mechanical stage fitting also will work. But the mechanical stage fitting will be a better choice for observing cells, blood cells, and the structure of blood cells. That's it. Coming to the observation. You will see three different types of corpuscles of cells. Red blood corpuscles. The red blood corpuscles will be most numerous. Most numerous. Each red blood corpuscle is enucleate. It is without a nucleus. The center looks relatively more transparent than the margin. And sometimes the cells RBC are found to adhere onto each other, forming a specialized structure called Rolex. It's not shown here, Rolex. Rolex. This is an RBC. There will be another RBC, it will be adhering to it. Yet another RBC, yet another RBC. So 5 to 6 or 10, 12 RBCs will adhere onto each other by their sides, forming a structure called Rolex. Rolex is not here in this figure. Anyway, RBC, enucleate cells, most numerous cells. Then different types of WBC. There are different types of WBC. I mean, there will be neutrophils, there will be basophils, eosinophils, lymphocytes, monocytes. These are less numerous. Less numerous. 600 times less numerous than RBC. Remember, the RBC WBC proportion or ratio is 600:1. RBC 600 numbers to 1 only WBC. 600:1. Then there will be platelets, but the platelets are not visible under the magnification that we will have an access to. We do not have an access to very higher levels of magnification. Only to some extent we have an access to magnification. At that magnification, we will not be able to see the blood platelets. A higher magnification is required, and the blood platelets will be visible as very small ducts scattered here and there on this in the smear. Now, this is all about the experiment. Next experiment is yes, catalase activity. We are taking up UV catalase activity next experiment in succession. Demonstration of catalase activity in human saliva. Let me tell you a little about the catalase enzyme. It is an enzyme. It's a beneficial enzyme. Beneficial in the sense it helps human in a specific way. It catalyzes the breaking of hydrogen peroxide into water and molecular oxygen. Remember, water and molecular oxygen in the presence of catalase. But if catalase were not there, if there will be no catalase, hydrogen peroxide will dissociate into water and nascent oxygen, atomic oxygen. Atomic oxygen or nascent oxygen is very reactive. It can react with quite a number of macromolecules of the body and change their structures. This is harmful. So, catalase enzyme, what it does, it prevents the dissociation of hydrogen peroxide into water and nascent oxygen. Rather, it promotes or encourages or catalyzes the conversion of hydrogen peroxide into water and molecular oxygen. So this molecular water is neither harmful nor is molecular oxygen. Both are beneficial. So catalase activity is beneficial for the body. Let's see where it is present. It is present in saliva, blood, and many extracellular fluids of the body. We now will test the presence of catalase and its activity in human saliva. A working principle I have already, let me tell you about the working, a little about the working principle. In order to test the action of catalase or catalase activity in human saliva, we what we do, we take hydrogen peroxide in a test tube, 3% hydrogen peroxide solution in a test tube. Ma'am, please give me a test tube. You take 3% hydrogen peroxide in a test tube. To it, you add paper discs. Paper disc means here is a punching machine. Look at this. These are paper discs. Please focus. These are paper discs. These are punched from a Whatman filtered paper. These are paper discs. Small paper discs. We can have these discs by punching the paper with a punch machine. Everybody must have seen a punch machine. So, a paper disc is held with a forceps. Then it is soaked with saliva. See, saliva completely soaked. You have some saliva from the mouth. Soak the paper disc You have already taken 3% hydrogen peroxide solution in this test tube. You drop it. Madam will demonstrate the experiment. I am going through its principle. You drop it. The paper disc has saliva. Hence it has catalase enzyme. Here in the test tube we have hydrogen peroxide solution. This catalase will catalyze the conversion of the hydrogen peroxide into water and molecular oxygen. The molecular oxygen that is evolved will cling on to the paper disc. Hence, the paper disc will start moving up the column. This is the column of hydrogen peroxide solution. It will start moving up the water column and will float finally on the surface. This indicates that molecular oxygen has evolved by the catalytic activity of catalase present in the saliva. Okay? Ma'am will demonstrate this experiment. It's a very fantastic experiment, easy experiment to undertake. The result she will show to all of you. Madam has taken 3% hydrogen peroxide solution in a test tube. Okay. She will now drop a saliva soaked. Very simple. There is no technicality at all in this experiment. Saliva soaked. Okay. It's dropped already. She has dropped a saliva soaked paper disc. What will be the consequence? The consequence is the catalase present in the saliva will catalyze the breaking of hydrogen peroxide into water and molecular oxygen. What will happen to the evolved molecular oxygen? That will cling on to the paper disc. It will slowly rise up the column and will finally float on the surface. Very interesting. While after say 2 to 3 minutes time, this will come to the surface. How does how is catalase beneficial to human? It catalyzes the breaking of hydrogen peroxide into water and molecular oxygen. If catalase were not there, in the absence of catalase, hydrogen peroxide must be broken down into water and nascent oxygen, atomic oxygen. Atomic oxygen is a harmful atom, very harmful. It can react with many macromolecular constituents of the body, and it could change the structure and configuration of these macromolecules. This is quite detrimental for the body. And of late, now it has been, people have been telling that, scientists have been telling that hydrogen peroxide that releases atomic oxygen may be suicidal for the body, Some cells or cells and mass will die due to the release of nascent oxygen. However, this is countered by the presence of catalase. So catalase prevents a mass death of cells. Okay, you can see that's here. You can see the focus. The paper disc was saliva soaked. Put in hydrogen peroxide, 3% hydrogen peroxide solution. It went up, came to the surface. Why? The evolved oxygen clung down to the paper disc and help it move up the column and come to the surface. A very simple experiment. There is no technicality at all in this experiment. Okay, demonstration. Okay, that's it.